The main way that our bodies and the bodies of all those other animals and other organisms out there, most organisms out there, generate the bulk of their energy is through oxidative phosphorylation. In this process, electrons are passed from something that wants it less, so something with a lower reduction potential, to something that wants it more, something with a higher reduction potential. This is energetically favorable, so you use the energy that's released in these pass-offs to power the pumping of protons, creating a proton gradient that is then used to, like a hydrolytic dam, turn the turning of this ATP synthase protein and generating ATP. This process, therefore, it takes the electrons from your carrier like NADH or FADH2, passes them off ultimately to oxygen, and you generate ATP. We saw how one of the sources of the NADH was glycolysis, so the breakdown of glucose. In glycolysis, we generate 2 ATP on net directly, as well as these 2 NADH. These 2 NADH are what are actually going to give us the bulk of the energy from the glucose, from the glycolysis part of it at least, but we need to take those electrons into the electron transport chain. There's still a lot of energy held in the pyruvate that's at the end of glycolysis, and so next we're going to later look at how we can actually take that pyruvate further, how we can bring it through the tricarboxylic acid cycle, the TCA, the Krebs cycle, citric acid cycle, there's multiple names for it, but you can see that we're generating a lot more of our NADH as well as our FDH-DH2, which is actually happening in that C2, that complex 2 of the electron transport chain. This is the same thing as that. That's that actual step. So that's your complex 2. And so we're going to get much more into that. But first, let's start with what happens with the NADH that you generate in glycolysis and what happens if you can't go through the electron transport chain. Remember that the electron transport chain was depending on the presence of oxygen to serve as this last electron acceptor in the chain. If you don't have oxygen, you can't pass here. Everything is going to back up. You're not going to be able to pump protons. And if you can't pump protons, then you can't make ATP. If you want to, however, keep glycolysis going in the absence of oxygen or the absence of sufficient oxygen, or for any other reason you can't do the electron transport chain, like if you're an erythrocyte or a red blood cell, those don't actually have mitochondria, so they can't go through the electron transport chain. Instead, they rely solely on glycolysis. So your red blood cells are kind of just little minimized sacs where the big thing that they do is make hemoglobin, and so that they can carry oxygen throughout the bloodstream. If that's their only function, then they don't really need mitochondria, and so they don't even need the nucleus, and so they get rid of all of that stuff and mainly just focus on making protein, and then they get the liver to do the hard work for them, and we'll talk about that in a minute, but they're going to rely only on glycolysis. We also see glycolysis be predominantly used in our muscles when we're working really hard, as well as various other cells will rely on glycolysis heavily. So yes, in glycolysis, you generate that 2 ATP on net, but you also are generating NADH. Normally, we think of that NADH as a good thing because it can go through the electron transport chain. But what's going to happen is if you can't go through the electron transport chain, that NADH is going to build up and you're going to run out of your NAD+. There are a couple of different problems with this. One is that you need that NAD+ in order for glycolysis to continue. NAD+ is used in this glyceraldehyde 3-phosphate dehydrogenase step as a cofactor, and it's kind of like one of your reactants where basically you need the NAD+ to pass the electrons onto to get your NADH. If you don't have that NAD+, you can't do that reaction. Normally, we keep NAD+ in a high ratio in proportion to NADH. This makes it so that NAD+ is going to be a good source for when we want to oxidize things. By keeping it in this high NAD+ NADH ratio, we're able to make it thermodynamically favorable to pass electrons to NAD+ from our carbon sources and then have that NADH that's generated go down through the electron transport chain. The power of the pass-off, the amount of energy you get from the pass-off, is going to depend in part on that NAD plus to NADH ratio, because the bigger that ratio is skewed, the more out of balance it is, the more energy you'll get when you get towards its equilibrium. So when you pass electrons to NAD plus, when it's further from equilibrium, you're going to get more energy. And so that's going to be more thermodynamically favorable, and that's going to help drive the reaction forward. If that ratio becomes lower, things get less favorable, but things also are getting less favorable in the presence of high NADH, because NADH is actually going to directly inhibit catabolic reactions. So remember, catabolism is the breakdown of molecules for energy typically, and catabolism is typically associated with oxidative reactions. When we want an oxidative reaction, we want that NAD plus, and we don't want that NADH. If we have a lot of oxidation happening, if we're building up NADH faster than we can recycle it, that could be a signal to our cells that maybe we should go and we should do anabolic things. So maybe we should start building molecules. And so slow down on the breakdown. And so let's actually directly use NADH as an inhibitor to inhibit catabolic enzymes. We'll talk later about how NADH acts as an allosteric modulator of glycolysis and of the citric acid cycle. So we'll talk about regulation of glycolysis after we talk a little about hormones and tissue-specific metabolism concepts. But we'll see that NADH can actually directly inhibit pyruvate kinase, so that enzyme that takes phosphoenolpyruvate to pyruvate, that big bang at the end of our glycolysis. And it's also going to inhibit pyruvate dehydrogenase, which is basically going to take that pyruvate and turn it into acetyl-CoA that can then enter the tricarboxylic acid pathway. So we're going to have NADH directly inhibit that complex, as well as inhibit our pyruvate kinase. And then we'll also see that it's going to inhibit multiple steps of the citric acid cycle. Bottom line is, if we're building up our NADH, we're going to be inhibiting catabolism. If we want catabolism to continue, we're going to need to regenerate that NAD plus and get rid of that NADH. There are several ways in which we can do this. The way that we've talked about already is by regenerating that NAD plus through the mitochondrial electron transport chain. In this process, we're going to be passing those electrons off from our NADH, and in the process, we're actually going to be generating ATP. Therefore, this is going to allow us to get the energy out of that NADH, get the maximum energy from whatever molecules we're breaking down, and regenerate NAD plus in the process. But as we mentioned, that's going to require the presence of oxygen and the presence of mitochondria. If we don't have those things, then we can't do this aerobic respiration. If we can't do aerobic respiration, there are options to anaerobically ferment. So anaerobic in the absence of oxygen, we can do this process called fermentation. We can also do it in the presence of oxygen, but it's not really as useful in the presence of oxygen because, well, then you're not getting the energy that you could be getting by taking those electrons through the electron transport chain. We'll talk about how there's a bit of a complication when it comes to actually getting electrons to the electron transport chain. But for now, just know that the electron transport chain is going to be an option most of the time. And then we have this option of anaerobic fermentation. When we talk about fermentation, we're typically talking about anaerobic conditions where basically we're regenerating NAD plus, so we can keep this cycle going without using electron transport chain and oxidative phosphorylation and all that good stuff. In our bodies, we do lactate fermentation, and we'll get a lot more into this process. There are also organisms that do alcohol fermentation and that are actually able to make ethanol from that pyruvate they generate from glycolysis. And in that process, they're going to regenerate the NAD plus in order to keep glycolysis going. What we'll see in common with all of these things is that we're having some sort of reduction reaction after we end glycolysis. So remember, glycolysis is an oxidative reaction. And so if we want to regenerate that NADH plus, we're going to need to oxidize the NAD, which means we need to reduce our substrate. So we do some sort of reduction reaction to that pyruvate in order to regenerate our NAD plus. So this is showing it in the stoichiometry, like per 1 glucose, you get 2 pyruvate. In some bacteria and yeast and plants, they go through this alcohol fermentation. They take that pyruvate, they decarboxylate it, so they remove this CO2 group. This gives you acetaldehyde, and that CO2, well, that's what's going to make your bread rise and things, because that's going to escape as a gas. This acetaldehyde, so that step wasn't a reduction, but now you can reduce it with alcohol dehydrogenase, and this is going to give you your ethanol. We don't have this pathway, but we do have another option for fermentation. The option that we use is to make lactate. So just a quick note is that lactate and lactic acid, those are just the conjugate acid and conjugate base forms of the same molecule. So see both terms used, and they're just readily interconverted by giving or taking a proton. Remember that this giving and taking a proton is not going to be redox, because the oxidation numbers aren't changing. Remember that basically when you calculate oxidation state, you're thinking about how many of the electrons an atom kind of owns. And so the oxygen, because it's more electronegative than the hydrogen, it's basically owning these shared electrons already. And so if that hydrogen were to actually leave as a proton, well, it's leaving behind the electrons. Remember, a proton is just a proton, not an electron. And so it's leaving behind the electron, but it's like the oxygen already had that electron, so it's not going to change the oxidation state of anything if you have a deprotonation. So lactate to lactic acid is not redox. However, pyruvate to lactate is. More specifically, you're reducing pyruvate, oxidizing NADH in the process to regenerate your NAD plus that then you can use to power glycolysis. This is often used by muscles that are working really hard, so they don't have the oxygen that they need to take that NADH and take the electrons from it through this electron transport chain to regenerate ATP. And it's also going to be used by cells like your red blood cells, which again don't have those mitochondria and so can't do the electron transport chain. You wouldn't want that lactate to build up. Now, lactate, it's a myth that lactate actually causes your delayed onset muscle soreness, but it can inhibit your muscles in the moment if you're building up a lot of it. And also, you basically are going to be wasting a lot of fuel if you just have a bunch of lactate build up. Because remember that most of the energy we get from the breakdown of glucose is actually going to be happening because of the NADH and the VDH2 that we get after the steps of glycolysis. So we don't want to waste all that energy that's still stored in the pyruvate, or at this case, stored in the lactate. And so we're going to ship it off to that lactate off to the liver. When this happens from lactate from your muscles, we call it the Cori cycle, but something similar is happening with your blood cells and other cells that are doing lactic fermentation. What's going to happen is basically in your muscles, you're generating that lactate. And this is through this enzyme lactate dehydrogenase. So this LDH is converting your pyruvate to your lactate. This lactate can travel through your bloodstream. It's going to travel to your liver. In your liver, now you have another lactate dehydrogenase. So you had a copy of it in your muscles and a copy of it in your liver. Well, you have lots of copies of it. But basically here, you're going to run the reaction in reverse. So in your muscles, you were reducing pyruvate to make lactate and using NADH and regenerating NAD+. In your muscles, you're going to be taking lactate and you're going to be oxidizing it to form pyruvate. So this is taking NAD+ and generating NADH. This pyruvate then, if you wanted, you can go and use it to make ATP, or if you want to basically use this as a cycle. So remember, when we're talking about a cycle, we want to keep things going without having to add in more stuff. In this case, what we could do is we could take that pyruvate and we could actually use gluconeogenesis to make glucose. So remember that your livers have the machinery for doing gluconeogenesis, so for actually making glucose from pyruvate. And your other cells, except for like some parts of your kidney and stuff, they don't have the ability to do gluconeogenesis. So your muscles couldn't make fresh glucose from that lactate or from that pyruvate, but your liver cells can. In order to do gluconeogenesis, one of the things that you're going to need is that NADH. And so here we have the NADH where we need it for this glyceraldehyde 3-phosphate dehydrogenase step. So we've already generated the NADH that we need for the energy to power the making of glucose through this cycle. So we can keep that NADH out to basically use it as is if we want this energy balance. When we do gluconeogenesis, we're now able to make glucose. And yes, we're going to have to put energy in to make that glucose, but we can do it in the liver, and it's still worth it because we get this glucose that we can then ship back to the muscles, giving the muscles a fresh source of energy so they can keep going. So to summarize, we have glucose. It's good. Glycolysis is happening in your muscle cells and generating pyruvate. We're also generating 2-ATP that the muscles can be using directly, and we generate this 2-NADH. The NADH, when there's not enough oxygen with your muscles working so hard, the NADH can't go through the electron transport chain, but we can recycle it to NAD+ by using it to reduce pyruvate to form lactate. That lactate can then get shipped to your liver. Your liver will then use that lactate, take the lactate dehydrogenase in the liver, oxidize the lactate to pyruvate, regenerating our NADH. This pyruvate and the NADH can then be used in gluconeogenesis to make glucose, and that glucose can then get shipped back to your muscle cells. Similar things are happening if that lactate comes from red blood cells or any other cells. They can all enter the liver where they can get converted into glucose that can get sent back to your muscle cells. Going back to when we talked about kind of like the different routes of gluconeogenesis, and we talked about how they were kind of two routes in terms of which reaction you did in the mitochondria and what you did in the cytoplasm when it comes to those first steps of reversing glycolysis, so your pyruvate to oxaloacetate and your oxaloacetate to oxylenol pyruvate, or going to malate, and then malate to oxaloacetate in the cytoplasm. Basically, what's going to happen is if you have lactate come in, you're going to regenerate the NADH in the cytoplasm already. And so then typically what you're doing is you're going to take that pyruvate and convert it to oxaloacetate and phosphoenol pyruvate in the mitochondria and then ship that out. Whereas if you are taking pyruvate from alanine or from just pyruvate, you typically are going to do just the oxaloacetate to malate step. Then the malate is going to get shipped out. When you ship out the malate, then you can use malate dehydrogenase in order to oxidize the malate to oxaloacetate. And here you're generating the NADH in the cytoplasm. You didn't need to do that step when you went in through lactate because here you're already generating NADH in the cytoplasm. I'm bringing this up now because this malate dehydrogenase step is something we're going to look at in more detail in a different context. So with malate dehydrogenase, we're taking malate and we're converting it to oxaloacetate. And this is an oxidation reaction. So we're taking NAD+ and we're regenerating NADH. We use this in a process called the malate aspartate shuttle. To review, if we want to keep glycolysis going, we need to regenerate NAD+ and get rid of NADH. If we want to get the most energy, we want to aerobically respire. We want to take those electrons from our NADH and pass them through the electron transport chain, generating ATP. In order to do that, however, we're going to need oxygen. If we don't have oxygen, the electrons can't get passed. If the electrons can't get passed, protons can't get pumped. If protons can't get pumped, ATP can't get made because you can't make your gradient to power the synthesis of ATP. Therefore, there are mechanisms by which we can regenerate NAD+ without oxygen present, that is anaerobically. We call these methods fermentation in our bodies, we do lactate acid through fermentation. Basically, what we do is we use lactate dehydrogenase in order to reduce pyruvate to lactate. That lactate can then basically get shipped to the liver, and the liver can convert it back to pyruvate. So it can oxidize it back to pyruvate, which can then go through gluconeogenesis in your liver to make glucose, which can then get shipped back to the muscles that need it in what's called the Cori cycle. Something similar is happening in your blood cells and in other cells that don't have mitochondria and therefore need to ferment. In yeast and plants and some microbes, they can ferment via ethanol. So they'll use pyruvate decarboxylase to basically decarboxylate pyruvate to form acetaldehyde and then reduce the acetaldehyde in order to form ethanol. One way or another, we're regenerating that NAD+, which is important so that we have it to do glycolysis, because remember, we needed it for a step in glycolysis, and it also is going to prevent it from inhibiting our catabolism. So we'll talk a lot more about how NADH can serve as an allosteric regulator of enzymes. So basically it binds to enzymes and inhibits their activity. More specifically, it's going to be binding to catabolic enzymes, so enzymes that are responsible for breaking down molecules for energy. We have a lot of NADH building up. This is kind of indicating that we're doing a lot of breakdown and maybe we need to slow down. Maybe we want to do some anabolism and start building things up.